The entire duration of pregnancy is about 280 days or 40 weeks or 9 months plus minus 7 days. And this pregnancy is divided in three periods. First is the germinal period, which extends from fertilization to third week of intrauterine life. Then the embryonic period extends from fourth to eighth week, and the fetal period extends from third month onwards up to the termination of pregnancy. So today we will discuss about the changes occurring in fertilized ovum in first week of development. So this first week of development is the journey from fertilization to implantation. So once the male and female pronuclei fuse, the process of fertilization is completed, then it undergoes cleavage division and it forms a stage called morula. Then there is formation of blastocyst, and finally the blastocyst gets implanted in the uterine cavity. So let us discuss the first week of development in detail. After fusion of male and female pronuclei, a large zygote is formed. A single cell stage zygote is formed. Now we know that the zygote is very large, it is huge, and in order to bring it to the normal size, there is segmentation of the zygote into small blastomeres by repeated mitotic division, and this process is known as cleavage division. In case of human beings, the fertilized ovum has very scanty deutoplasm. That's why the whole ovum is involved in cleavage division. So it is known as holoblastic cleavage division. As the cleavage division starts, initially we have a two-cell stage that appears at about 30 hours after fertilization. Further, these cells will undergo mitotic division and it's converted into a four-cell stage at about 60 hours. Then, further mitosis causes the 12 cell stage, means we can see small segments known as blastomeres. So 12 cell stage reaches by about 72 hours after fertilization. 16 cell stage appears at 96 hours after fertilization. And when this fertilized ovum moves from the ampulla towards the uterine cavity, it is of about 12 to 16 cell stage. As it enters the uterine cavity, the uterine milk will enter into it and it is soon divided into outer cell mass and inner cell mass. After that, the uterine milk will enter into the fertilized ovum, or the means when it is of 12 to 16 cell stage, it is known as morula. After that, a cystic cavity appears. Small cavities are there, they will coalesce and they will form a large cavity known as blastocele, which is filled with fluid, and soon this will be converted into blastocyst, which has outer cell mass, which will form future which is known as trophoblast, and it will form future chorion. Then the inner cell mass, it will form embryo proper, and the blastocele is the cavity which is filled with the fluid. So this is the blastocyst and it is covered by zona pellucida. During cleavage division, when the zygote reaches 12 to 16 cell stage, it is known as morula. So this morula is covered by zona pellucida, and by the process of compaction, the outer trophoblastic cells are tightly packed by desmosomes and gap junction. So the outer cell mass is known as trophoblast, which will form future chorion of placenta, and the inner cell mass forms the embryo proper, so it is known as embryoblast. When the cleavage division reaches 32 to 64 cell stage, it is known as blastocyst. Blastocyst appears at fifth day after fertilization. So, when the fertilized zygote moves from the uterine tube to the uterine cavity, the uterine milk will diffuse from the zona pellucida and it will get accumulated between these blastomeres, and small cavities are formed in which the fluid gets accumulated. Soon these small cavities will coalesce and they'll form a large cavity which is filled with fluid, and this cavity is known as blastocele. Okay, so we have the outer cell mass which is known as trophoblast, which will form the future chorion or placenta. Again, the trophoblast which is facing towards embryonic pole. Now what is embryonic pole? The side at which the inner cell mass is attached to the outer cell mass. So this side is the embryonic pole. So the trophoblast covering the embryonic pole is known as polar trophoblast, and rest of the trophoblast is known as mural trophoblast. So this is mural trophoblast and this one is the polar trophoblast. Then we can see the inner cell mass which is also known as embryoblast. It will form embryo proper, and the cavity is known as blastocele. Okay, the cavity is known as blastocele and it is filled with the fluid. And again, one more important thing is the blastocyst is covered by zona pellucida, which will prevent ectopic pregnancy. When the cleavage division is occurring simultaneously, the uterine endometrium also undergoes some changes. Now, the uterine endometrium is converted to decidua. So, this decidua is more receptive for implantation. The uterus is in secretory phase, and in the decidua, there is deposition of more glycogen lipids in the stroma. So this decidua is again divided in three parts. The deepest one or the outermost is the stratum basal, then there is stratum spongiosum, and facing towards the luminal side is the stratum compactum. So uh, now after the formation of decidua, the zona pellucida covering the blastocyst will disappear by about fifth day, and it is by the trypsin like enzyme. The process is known as hatching. So the blastocyst is hatched from the zona pellucida, and this blastocyst has a covering of trophoblast. This trophoblast has proteolytic activity, or this trophoblast will help the blastocyst or developing embryo to get embedded within the uterine endometrium. So the trophoblast facing towards the endometrium is known as polar trophoblast, and it will start eroding first the stratum compactum, then it will get embedded within the stratum spongiosum. It is completely embedded in the uterine wall. So the process of implantation starts by 7th day and it is completed by 12th day. So the embryo now receives oxygen and nutrition from the mother side. Normal site of implantation is the upper segment of the uterine cavity. So it is at the junction of posterior wall of the uterus and the fundus. Junction of fundus and posterior wall of the uterine cavity is the normal site of implantation. It occurs in upper segment of the uterine cavity. Now, what is ectopic site of implantation? When there is abnormal implantation, or it is implanted at a site other than upper segment of the uterine cavity, then it is ectopic implantation. So if it can be intrauterine or extrauterine ectopic pregnancy. Intrauterine ectopic pregnancy is the one which is occurring in the lower segment of the uterus, and it is known as placenta previa. There are four grades of placenta previa. When the implantation occurs in the lower segment, it is called first degree of placenta previa. When the implantation reaches up to the internal os, it reaches up to the internal os of the cervix, then it is called grade 2 placenta previa. Grade 3 placenta previa is the one where implantation occurs in at the level of internal os, but when the cervix dilates, then the os will open. And grade 4 placenta previa is the one in which the implantation occurs in the lower segment. It is completely covering the internal os. Even when the cervix is dilated, it will remain closed. So these are the four grades of placenta previa which will occur if the implantation takes place in the lower segment of uterine cavity. Now what are the extrauterine sites of ectopic pregnancy? So the first and most common site of ectopic pregnancy is tubal pregnancy, which occurs in the fallopian tube. Apart from that, abdominal pregnancy, then we can see the ovarian implantation also. So these are some of the intrauterine and extrauterine sites of ectopic pregnancy. Slide we can see two clinical conditions. So the first slide is of ectopic pregnancy, mainly the tubal pregnancy. When the implantation occurs in the fallopian tube, it is known as tubal pregnancy, and the complication is it may rupture. Then the second slide shows Hydatidiform mole. So what is this Hydatidiform vesicular mole? It is a clinical condition which occurs due to abnormal development of trophoblastic cells. And when the fertilization is occurring, the oocyte is absent or it is inactive pronucleus. And this is fertilized by a sperm, or due to the fertilization of empty oocyte with two sperm. It is known as dispermic mole. So this leads to the death of embryo, and as the trophoblast development is abnormal, the chorionic villi fail to complete their development and they undergo degenerative changes. And the degeneration of these chorionic villi leads to formation of cystic swellings known as Hydatidiform mole. It resembles bunch of grapes, and when these moles are present, they secrete high level of HCG, human chorionic gonadotrophin. And in the female, we can see some clinical features like there is preeclampsia during first trimester, there is a raised HCG level, then dark brown or bright red vaginal bleeding is there. These hydatid moles, they resemble bunch of grapes, so the vesicles are present in the urine. And most important, there is no fetal movement, there is no fetal heart sound, and ultrasound typically shows, means important for MCQ also. So the ultrasound reveals snowstorm appearance of the uterine cavity, and these hydrated form moles are more prone to malignant changes, and the malignancy of this hydrated form mole is known as choriocarcinoma. Hydatidiform mole can be complete when there is no existence of embryo, or it can be partial in which a part of embryo is seen. Okay, so this is about hydatidiform mole. Why is the cleavage division important? What is the significance of cleavage division? So the cleavage division brings about distribution of cytoplasm of the zygote among the blastomere. It facilitates morphogenetic movement for cell differentiation, germ layer formation, formation of tissues and organ. Also, it restores normal size of the cell and normal nucleocytoplasmic ratio, which is characteristic of species. Then the unicellular zygote is converted into multicellular embryo by cleavage division. Let us briefly revise the events occurring at the first week of intrauterine life. So, initially, the secondary oocyte arrested in metaphase of second meiotic division is ovulated. It will reach the ampulla. It will get fertilized with the sperm. As soon as fertilization occurs, the second meiotic division is completed. The second polar body is released in the perivitelline space. The zygote is formed, which is covered by zona pellucida. After 30 hours, first mitotic division takes place. A two-cell stage is reached. Then further, mitotic division reaches four-cell stage at about 60 hours. Then the 8-cell stage, then 16-cell stage. So it is known as morula. It is a solid ball of cell. So these cells formed by the segmentation of zygote are known as blastomeres. By the process of compaction, outer trophoblast or the outer cell mass will form the trophoblast and the inner cell mass is forming the embryoblast. Now when this morula reaches the uterine cavity, the uterine fluid will enter in it and the small cavities will fuse and they will form a large cavity. So a large cavity is formed which is known as blastocele. So there is a blastocele. The outer cell mass will form the trophoblast which will form the chorion of the placenta. The inner cell mass will form the embryoblast and it is attached to the embryonic pole. So this is the embryonic pole. So the inner cell mass, outer cell mass and the blastocele will form the blastocyst, and this blastocyst is initially covered by zona pellucida. At fifth day, by the process of hatching, the zona pellucida is removed or it disappears by trypsin-like enzyme, and the blastocyst gets implanted at the normal site, that is at the junction of fundus with the posterior wall of the uterus. Then the proteolytic enzyme present in the trophoblast will help in the erosion of the blastocyst in the uterine endometrium, and it will then the blastocyst will then get completely embedded within the stratum compactum and stratum spongiosum of the deciduum of uterus, and after implantation the site from where it is entering the cavity will be covered by fibrin plaque. So implantation occurs or it starts at 7th day after fertilization and it is completed by about 12th day of after fertilization.